Classroom setting with professor standing in front of a whiteboard. Professor: Good morning, everyone. Today, we are going to dive into the fascinating world of molecular biology. Specifically, we will be exploring the intricate mechanisms of CRISPR Cas systems and their applications in various fields. Students respond with enthusiasm. Professor, before we delve into the details, let's begin with some basics of molecular biology. Professor, understanding the CRISPR Cas system, a defense mechanism in bacteria against viral infections, is crucial for medical advancements. This system uses CRISPR RNA and Cas proteins to target and destroy viral DNA, offering potential applications in gene editing and targeted therapeutics. CRISPR Cas systems are a defense mechanism in bacteria. They incorporate foreign DNA into their own array, producing small RNA molecules called CRISPRs, which guide Cas proteins to degrade and inactivate viral DNA. These systems have revolutionized genetic engineering and offer promising applications in medicine. Professor, the CRISPR array is made up of repeated DNA sequences separated by unique spacer sequences. These spacers store the memory of past viral infections. When a bacterium encounters a new viral threat, it captures a piece of the viral DNA as a spacer and integrates it into its CRISPR array. This process, called spacer acquisition, helps the bacterium remember and defend against future viral attacks. The professor explains the role of Cas proteins in antiviral defense. Professor, in the context of antiviral defense, Cas proteins play a crucial role. When a viral DNA is detected, Cas proteins are programmed to bind and cleave the viral DNA, rendering the virus unable to replicate and infect host cells. This mechanism provides a powerful defense against viral infections. In the CRISPR Cas systems, the Cas protein RNA complex scans the foreign genetic material for matching sequences and binds to them, forming an OR loop structure. The Cas proteins then cleave the foreign genetic material at specific sites, disabling viral or plasmid DNA. The CRISPR Cas system continuously acquires new spacers from foreign DNA, enhancing its ability to defend against evolving viral threats. CRISPR interference effectively acts as a potent antiviral defense mechanism. CRISPR Cas systems help bacteria defend against viruses by integrating new genetic sequences, called spacers, into their CRISPR array. This adaptation process, facilitated by Cas proteins, creates immune memory and enhances bacteria's ability to fight future viral infections. The scene shifts back to the whiteboard. Professor, while CRISPR Cas systems are highly effective in defending against viral pathogens, it is important to note that viruses can also evolve and adapt to overcome this defense mechanism. The professor talks about viral countermeasures and strategies to bypass CRISPR interference. Professor, some viral escape strategies involve mutations or deletions in the protospacer sequence, preventing efficient recognition by the CRISPR Cas system. Additionally, some viruses produce anti-CRISPR proteins that inhibit or neutralize Cas protein activity. The professor emphasizes the role of PAM sequences in viral defense. Professor, understanding PAM sequences is essential for the CRISPR Cas system to effectively combat viral infections. These short sequences act as molecular flags, enabling Cas proteins to specifically target and destroy foreign DNA while sparing the host genome. The scene shifts back to the whiteboard. Professor, CRISPR Cas systems not only interact with foreign genetic material but also have intricate relationships with the host cell machinery. The professor discusses the interactions between CRISPR Cas systems and the host cell. Professor, the expression of CRISPR Cas components is tightly regulated, ensuring proper functioning and avoiding unnecessary damage to the host cell's own genetic material. Additionally, Cas proteins often interact with various cellular factors to mediate their activities. The scene transitions back to the classroom. Professor, CRISPR Cas systems were initially discovered in bacteria, but they have also been adapted for use in eukaryotic organisms, including plants and animals. The professor explains the challenges and advancements in applying CRISPR Cas systems in eukaryotes. Professor, in eukaryotes, the delivery of the CRISPR Cas components to the target cells and the efficient DNA cleavage are among the critical challenges. However, significant progress has been made, opening up possibilities for greater precision in genetic engineering and disease treatment. The scene shifts back to the whiteboard. Professor, the application of CRISPR technology extends far beyond fundamental research. It has revolutionized genetic engineering techniques and opened up new possibilities in various fields. The professor discusses different applications of CRISPR technology. Professor, some of the notable applications include gene editing, gene therapy, agricultural improvements, and even disease diagnostics. CRISPR technology has the potential to reshape our understanding of biology and revolutionize medicine. The scene transitions back to the classroom. Professor, as with any powerful tool, the use of CRISPR technology raises ethical and societal concerns that must be considered. The professor explores the ethical implications associated with the use of CRISPR technology. Professor, questions surrounding human germline editing, potential unintended consequences, and equitable access to CRISPR-related therapies are among the ethical considerations that must be carefully addressed. CRISPR Cas systems have transformed molecular biology, allowing precise genome editing with potential healthcare breakthroughs. Understanding their principles, applications, and ethical considerations is crucial for emerging medical professionals.